Hi, my name is Masaru Kanekio. I'm from Vaccine Research Center, NIAID, National Institutes of Health. And thank you for organizers for giving me the opportunity to present our work in this microbiology virtual week. So today I'm going to talk about nanoparticle-based next-generation influenza vaccines. So before we start, this is my public disclosure statement. And I want to start with this slide, highlighting some of the technology breakthroughs in the mid-century and their evolution afterwards. So showing on the left is the first microwave developed in 1947. It is called laser range. It is 6 feet tall, 800 pounds, and cost more than $50,000. Around the same time, they start developing influenza vaccines using embryonated eggs. So shown here in the picture is one of the picture from 40s, I think, infecting embryonated eggs with influenza viruses. And as you all know, that microwave today is less than 1 foot tall, less than 20 pounds, and costs less than $50. But today, still the influenza vaccine production is heavily relying on embryonated egg and the technology developed over 75 years ago. So we believe the technology can revolutionize the influenza vaccine in the near future. So here are the technology focus at the VRC influenza vaccine program. So we have 5 Ds. So the first D is discovery. So this is by isolating and characterizing monoclonal antibodies to influenza virus, and hopefully to identify the viral sites of vulnerability that inform the vaccine design. So the second D is the design. So we are heavy on the structure-based and structure-guided protein engineering for vaccine immunogen as well as research tools. And third one is display. So this is something that we are investing a lot of time and efforts in these years to find the best format to display antigens to the immune system. So we use both natural and designed nanoparticle platforms to do that. And fourth one is the delivery. So this is traditionally our vaccine is protein-based, but now we know that other options such as messenger RNA is a pretty attractive tool for the vaccine delivery. So that is also in consideration. Adjuvants is another one, and also routes of immunization. The fifth D is the detection. So we are developing assays and tools for specific immunological endpoints. So this includes the precise serological assays, single-cell omics, to identify and to delineate immune responses to influenza vaccine. So influenza viruses, 2 major proteins on the surface. 2 of them are the glycoproteins on the surface. So one is shown in blue here is the hemagglutinin, HA, and another one is called NA, so the neuraminidase. So for the sake of time, I don't go through the NA-based design, but this is something more in the focus in these days. But HA is a major vaccine-induced immune response target. So the HA is a homotrimeric protein, which has structurally defined head domain and stem domains. So the head domain is a hypervariable domain of the HA, so which differs strain by strain, but it's immunodominant, so it's a very immunogenic protein. And the stem domain, in contrast, is a highly conserved domain, which includes the virus fusion machinery. So this domain is usually immunosubdominant, but antibody against this domain can be very broadly cross-reactive. So we have 3 different approaches and 3 different antigens. So we made nanoparticle vaccines of using the receptor binding domain, which is a part of the head. So by displaying them as a mosaic array on the nanoparticle, shown here on the top, the mosaic RBD nanoparticle, and shown in the middle is the HA nanoparticle, which displays a trimeric exodomain of hemorrhagine on the surface of self-assembly nanoparticles. So this self-assembly nanoparticle is the basis for our next generation vaccine, and it can mount the various trimeric glycoproteins on the surface. The bottom here is called HA stem nanoparticles. So this is displaying, instead of full-length HA trimer, it's displaying engineered stabilized stem nanoparticles, or stem trimers on the surface. So each have different goals to achieve better protection against ever-changing influenza viruses. So the first approach, the mosaic RBD nanoparticles, is avoiding immunodominance. So this is displaying multiple antigenic variants at the same time that promote the immune system to recognize the conserved domain among the displayed antigens. So this will be a good way to avoid immune dominance and also overcome the antigenic competition between the antigens. So the HA nanoparticle this is a highly immunogenic form. So we can generate immune response, very potent immune response to the HA. So by doing this way, we can quickly generate vaccine, and hopefully we can accumulate the breadth by combining multiple different HA nanoparticles. So the stem nanoparticles are displaying the conserved domain of HA. So the goal is to target the immune system to generate antibodies against that the conserved site, so the sites of vulnerability of the virus. In the later of today's talk, I'm going through some newer technologies, including this mosaic HA nanoparticle, so which have a full-length HA trimer on the surface of nanoparticles, but multiple different kinds of HA at the same time. So starting with this self-assembly nanoparticle platform, we developed now like several years ago using these Heterobacter pylori-based nanoparticles. So this forms an octahedral particle with the 24 identical subunits. So this platform is assembling in vivo, which means upon biosynthesis of the protein, they spontaneously self-assemble into a particle. By using this platform, we and others have been very successful for generating vaccine candidates, not only against influenza virus, but also against herpes virus, Epstein-Barr, RSV, and more recently, Lyme disease. So, but we focus more now today on the influenza virus. So I'm going to show you a couple of progress that we made in the last several years. So first, the HA nanoparticles. We reported in 2013 about the development of the vaccine. In 2017, we initiated the human phase 1 clinical trial with the prototype HA nanoparticle vaccine. And now the trial is fully enrolled, and we almost finished analysis. So we are hoping to report the results from the trial soon. And a few years after, we developed the stabilized stem nanoparticles using the group 1 H1 subtype HA. So this vaccine generates broadly cross-reactive stem-directed antibody responses that is protective against heterosceptic virus changes in animal models. So we initiated the phase 1 clinical trials at VRC in 2019, and the trial is still ongoing, but hopefully we can report the results from the trial anytime soon. So shortly after the group 1 human trials began, we published a paper about group 2 HA stem nanoparticles. The group 2 stem nanoparticles are not very easy to make. So at the VRC, we spent several years to design and optimize the stabilized stem immunogen for the group 2 HA. And shown here is the cartoon showing all the mutations we made to stabilize that HA stem domain from the group 2 into an intact trimeric structure. But we have made the stabilized HA and successfully displayed on the nanoparticles shown here. On the bottom is the cryo-EM structure of the stem nanoparticles of H10. So these group 2 stem nanoparticles are highly protective against heterosceptic group 2 virus challenges in animal model. So shown here on the right is that H10-based stem nanoparticle immunized mice challenged with different group 2 viruses, including H3N2, H7, and 9, in addition to H10N8, which is the homologous virus. Shown here on the bottom is the empty nanoparticle immunized control. So these H10 nanoparticles are now being produced at the BRC for intended phase 1 clinical trials starting this year. So now switching gear to a mosaic HARBD nanoparticle, which is we recently published. So I don't want to spend too much time on this because it's published, but let me introduce this concept about mosaic antigen display. So shown here on the left, this is a very simplified view when the immune system encounters the particulate antigen, including viruses and vaccines. So each virus, even with a highly heterogeneous HAV or influenza virus, each virion have only one antigenic type on the surface. So shown here depicted as a blue antigen. So each particulate antigen, when encounters immune system, being recognized by B cells with the cognate BCR on the surface. So shown here in the middle is the blue antigen-specific B cells, which is recognizing the blue antigen on the particle. At the same time, we also have a subset of B cells that can recognize not only blue, but blue and some other antigens, which is also present, but it's outcompeted by the blue-specific response. But we hypothesized that if we introduce something non-natural heterogeneous antigen with multiple different antigenic variants on the surface, that kind of particles may interact very differently with the immune system compared to homogeneous antigens. So these heterogeneous mosaic antigens are having multiple different antigenic variants on the surface of the particle. So in this case, the blue-specific B cells have a difficult time to recognize it because the blue antigen on the surface of these mosaic particles are so diluted. And it's the BCR on these B cells can recognize only blue ones. But instead, these particles will be well recognized by the cross-reactive B cells, shown here in the bottom with the orange BCR. So these B cells can recognize not only blue, but also cyan, red, green, and yellow, for instance. So these B cells have now better opportunity to be expanded, propagated, and also contribute to the immune response. So this is our hypothesis, and to test this hypothesis, we developed the nanoparticle vaccines using HARBD, so which is highly divergent portion of the HA. So shown on the left is monotypic display with only single antigenic type on the surface. On the right is a mosaic display with multiple different antigenic types or antigenic variants on the surface of a single nanoparticle. So we can control this particle density and heterogeneity of the antigens by design. So the construct design, as I said, using HARBD, genetically fused to a ferritin subunit monomer, and then it self-assembles into a 24 metric particles. So we pick multiple different strains of HARBD and make nanoparticles individually presenting one antigenic type or as a mosaic display on the right. So the admixture is multiple different particles, each present single antigenic type. So as you can see, if you measure the immune response against the blue antigen, for instance, if you give admixture of multiple different particles, as you increase the valency of the antigen, you are getting reduced response to the blue. In contrast, on the right-hand side, if you present the same antigen in the mosaic antigen array, you don't see that kind of decreased response to the blue antigen, for instance. So this is a kind of new way to overcome this antigenic competition between the antigens by co-displaying multiple variants at the same time. So even more strikingly, when we immunize the particles, either cocktail, mosaic, or the sequential vaccination, where we immunize one or two particles at a time, but each time the animals see the different particles. So in that case, we expect the sequential vaccination also accumulate the breadth. But the mosaic nanoparticle outperforms either both the admixture of multiple different particles or the sequential vaccination. So shown here on the right is the global GMT of 5 different viruses tested. So the mosaic vaccination is outperforming other vaccination regimen. So to confirm that we are selecting the B cells that cross-recognizing or cross-reactive to multiple different antigens, we isolated monoclonal antibody from the mouse immunized with mosaic RBD nanoparticles. So shown here on the left is one of the example, 441D6 antibody in complex with HA trimer. As you can see, the antibody is shown here in the coral color. It's recognizing HA head. The recognition of the HA is, the site is different from the nucleic acid binding site, which is shown here in asterisk. So the 441D6 binding site is also different from the classically defined antigenic sites in the HA head. And now the antigenic site recognized by 441D6 is called the lateral patch of the HA. And this is highly conserved among different H1 subtype viruses. So indeed, when we test multiple different H1N1 viruses in the pseudovirus neutralization assay, 441D6 neutralize all of them. So this antibody is cross-reactive and induced by the mosaic RBD nanoparticles. And this is our working model of the mosaic nanoparticle vaccine. So when you immunize, when you stimulate the immune system with the homotypic stimuli, shown in gray, you enrich the immune population reacting with this gray antigen, and most of them are monospecific. But when you stimulate the immune system with the heterotypic stimuli, the mosaic antigen, you have much better chance to elicit the cross-reactive B cells by this type of stimulation. So that's the basis of the mosaic antigen display, and move on to the next one. So we have been playing a lot with this ferritin nanoparticle platform, but also exploring other platforms, including remote synthase, encapsulating, or even the bicomponent insect ferritin platform. But there are more flexible and advanced nanoparticle platform out there. So we started collaboration with the Institute for Protein Design at the University of Washington, especially the New King's group in Institute, who is pioneering this area of making those designed self-assembly nanoparticles. So we have been working together for years now to design a new influenza nanoparticle vaccine using his platform. So his platform is made of, shown here is a two-component nanoparticles, means that two different subunits make one particle. So shown here in blue is making trimer, and gray is making pentamers. And when you mix them together, it forms the nanoparticles because they made a specific interface between the trimer and pentamer that promote the self-assembly of those two subunits. So this design principle is very precise and controllable. So the beauty is one of the beauty of the system is that you can make pentamers and trimers in a separate reaction. So you can prepare your antigens in the different expression system, for instance, or different reactions. And once you purify the materials, you just simply mix them together to make particles. And this is ideal platform to make a controllable way of mosaic display. So we chose one of the nanoparticle platform, which has been developed by the University of Washington, have made of trimer and pentamer. So in this case, the pentamer is coming from East, and trimer is heavily designed by the protein engineering suite called Receptor. So the nanoparticle called I53DN5 is made of trimer and pentamer. And trimer, shown here in light blue, is designed optimized for the antigen display, the trimeric antigen display. So the small blue dot here is the N-terminus of the protein, so which is located near the threefold axis and then co-localized together in the cross proximity to accommodate the trimeric protein fusion. So, but when you mix this trimer and pentamer, it forms spherical nanoparticles. And shown here is the negative stain EM picture of the particles. And as you can see, the reconstruction image on the bottom left, the design is very accurate, and it produces a very homogeneous nanoparticle upon assembly reaction. So we use this I53DN5 platform to display HA, antigen from influenza virus. So in principle, we can display up to 20 different trimers per particle. But shown here as an example, at the bottom is the HA nanoparticle with the single HA. And the cryo-EM reconstruction image showing here is the nanoparticle with the spikes protruding from the core, and it's resembling the model. And when we isolate the spike density from the particle and then reconstruct based on the spike, based on the localized reconstruction it's made, HA displayed on the particle is very intact. And you can see the shape of the HA, and you can see all the antigenic sites are conserved, and the HA stem is available. So this is the ideal platform to display the viral antigens, especially in this case, the HA. So we move forward with this platform and then create the vaccine candidate. So we chose 4 different HAs to be present on the particle because these 4 are identical strains that of the commercial influenza vaccine of 2017. so which has H1, H3, and 2 influenza B HAs. So we made 2 preparations. So top here, we call cocktail I53B and 5. So each particle presents one antigenic type. For instance, the red one that is nanoparticle presenting H1 HA on the red one, and H3 HA on the blue one, and so on. And the second preparation we made is a quarter event mosaic nanoparticle. It is using the same component, but co-assembled into a single particles. So each particle has all 4 HAs on the surface. By doing the negative stain EM analysis, we can clearly see those well-formed core with proteolidin spikes projecting outward for those HA nanoparticles. And we did the immunogenicity study by using 3 different animal models, mice, ferrets, and non-human primates. And these are the 3 vaccines that we used in that study. So the first one is QID. So this is a commercial influenza vaccine from 2017-18 season. And the cocktail vaccine in the middle is the cocktail formulation that I just described, and the mosaic on the right. Animal has been vaccinated 3 times, and then we measured antibody responses after the vaccination. So here is the hemorrhagination inhibition titer after vaccination. So shown here is the vaccine matched and slightly mismatched influenza B responses in mice and ferrets and non-human primates. As you can see, the immune responses are induced by the HA nanoparticles as good as commercial vaccine, and sometimes you see a slightly better response by the HA nanoparticles. And here is the microneutralization titer, so which are a little bit more sensitive, I'll say, because you can detect not only the hemorrhagination inhibition activity, but also fusion inhibition as well. So the stem-directed antibody response can be measured in the microneutralization. So in this case, again, we see a robust antibody response, neutralizing response against vaccine-matched viruses in all 3 animal models. And a few cases, we see statistically higher responses by HA nanoparticles. So we are more interested to know about the breadth of the response. So shown here is the phylogenetic tree of H1N1 virus, the HA sequences on the left, and the phylogenetic tree of H3 sequences on the right. So we picked historical strains from both H1 and H3, and then analyzed the neutralizing response against those viruses. So as you can see, the HA nanoparticle immunized animals shown here in blue and red are showing slightly but higher neutralizing titer against those drifted viruses and than the commercial vaccines. So more importantly, when these immunized animals are challenged with the heterologous H1N1 or significantly divergent H3N2 virus, we see a better protection provided by the HA nanoparticle vaccination compared to QID vaccination. So not only the drifted strain, we are most interested to know about heterosceptic responses by those vaccines. So we picked a couple of different HA from both group 1 and group 2 influenza A viruses. So again, the H1 and the H3 are included in the vaccine, but nothing else is included in the vaccine. So when you look at the H5 and H6 response from group 1, you see a higher overall immune responses with the HA nanoparticle vaccinations compared to a QAD vaccination. And the same story is true for group 2 HAs. We tested H7 and H10, and we see significantly higher antibody responses against those two heterosceptic HAs. But importantly, these heterosceptic antibody responses are functional and providing protection in animal models. So here we vaccinated mice and challenged with either H5N1 on top or H7N9 virus on the bottom. So you can see the HA nanoparticle immunized mice are protected from death in this vaccination, in this challenge. But the commercial vaccine provides none or very weak protection against these virus challenges. And the trends are the same for the ferrets. So we challenged with H5 and H7, and we see a better survival as well as a lower viral load in the lung after a challenge. In non-human primates, we took a slightly different approach. So we purified polyclonal Ig immunoglobulin after vaccination. And those purified immunoglobulins we passively transferred into recipient mice and then challenged with the H5N1 or the H7N9. And you can clearly see that HA nanoparticle-derived IgG provides more complete protection against those heterosceptic viruses. So suggesting that protection we are seeing, the heterosceptic protection we are seeing is antibody-mediated, and the seasonal vaccine component is sufficient to induce this heterosceptic immune response. So to confirm that heterosceptic responses we are seeing is directing to the stem of the HA. And again, the HA stem domain is highly conserved across different subtypes. So we pick, we use the stem-only protein, which is developed at VRC by protein engineering. So we use both H1 or the H3 stem-only protein to detect the stem-directed responses. So as you can see, in all three animal models, we detect much higher antibody responses against the stem by HA nanoparticle vaccinations compared to the commercial vaccine. And it's most strikingly when we purify the polyclonal IgG from non-human primates and directly visualize with the cryo-EM, the technique called MPEM, so the epitope mapping by polyclonal electron microscopy. So we identified two major classes of antibody that recognize H5HA in those polyclonal IgG. And one of them is strikingly similar to a human antibody called MERI-8852, so which is one of the best antibody known so far against influenza viruses. So this is suggesting that non-human primates upon vaccination with HA nanoparticle can generate broadly cross-reactive stem-directed antibody responses that resembling the human responses. And strikingly, when we quantify the classes of antibody bound to the HA molecule by MPEM, we see a more diverse population of antibody classes against homologous antigen, H1 on the left, on the pie chart. But when you look at the H5 substrate, we only see the stem-directed antibodies. So suggesting that the stem domain is highly accessible in the vaccine and immunogenic. So to summarize the first part, I'm showing that self-assembling nanoparticles are powerful and highly flexible vaccine platforms. And first in human clinical studies proved that the nanoparticle vaccines are safe and well tolerated. And mosaic, and I introduced the mosaic nanoparticle display of oncogenic variants that can subvert immune dominance of strain-specific responses by providing a variety advantage to cross-reactive B cells. In the second part, I introduced the designed two-component nanoparticle platform that that allow us to do the in vitro assembly reaction of nanoparticle with programmable stoichiometry. So this is extremely useful for the designability of the vaccine. And using this platform, we made quadrivalent HA nanoparticles that are highly immunogenic in mice, threats, and non-human primates. And these nanoparticles elicit potent head-directed neutralizing antibody responses, as well as broadly cross-reactive stem-directed protective antibody responses. And based on this result, the VRC has initiated the GMP manufacturing of the quadrivalent HA nanoparticles and for the intended phase 1 clinical trials in 2021. And if you want to know more about quadrivalent HA nanoparticle, we have a preprint in the bioarchive. So with that, we see the next generation nanoparticle-based vaccines. I can do the simple multivariant display of antigens that can facilitate a better immune recognition, as well as the mosaic display, which will be selectively inducing cross-reactive B cells, and it's a better way to avoid antigen competition and immunodominance. And also, we are exploring the options to incorporating immunomodulatory molecules in the vaccine, so immune targeting, immune potentiation, and self-adjuvanting. And importantly, our nanoparticle designs are all based on the genetic fusion. So it's compatible with the genetic delivery, such as messenger RNA vaccine. With that, I'd like to thank many people involved in these studies, especially for Barney Graham, who directs the influenza program at the VRC, and in the clinical trial program, who leads the clinical trials of those proof of principle nanoparticle vaccines. And for the second part, the quadrivalent HA nanoparticles, the project was led by Seyhan Borger-Barnum from our group and Daniel Ellis from Neil King's group. So again, that project was very close collaboration with Neil King's lab at the Institute for Protein Design at the University of Washington. And it was my pleasure to do the work with them. So we have a lot more to report in the future. So I'm very excited. So I will stop here and thank you very much for your attention.